Hello everybody and welcome to my Canadian open neuroscience platform Lightning Talk. Today I'm going to be speaking to you about my project. It's called the Continuous Testing of Neuroimaging Results Across Pipelines and Datasets, and it has an application to hearing loss associations with brain structure. My name is Jacob Sands, and without further ado, let's get started. It is known that neuroimaging results are sensitive to variations in processing pipelines and that this can contribute to scientific result reproducibility issues. One good example of this is from this Bowring 2019 paper, whereby 3 different pipelines, AFNI, FSL, and SPM, were run on the exact same data with hopes of finding brain activations. And we can see that the brain activations for each of the 3 pipelines look slightly different, which is troubling from a reproducibility perspective when they're meant to be showing the exact same thing. On a larger scale, there was a study from 2020 that gave 70 different teams the exact same neuroimaging data to analyze, and this image here shows the correlation between the results of the different teams, and you can see that there are many colors in this correlation table, and it basically just shows that there wasn't much agreement, or there was a limited amount of agreement within all the teams. Similarly, there was a 2016 paper that showed that neuroimaging pipelines and the choice of pipelines that researchers enact can impact the interpretation of weakly significant results. So all of this to say, it can be hard to find a ground truth in this field, with all of the different disagreements between pipelines. A good example of methodological flexibility in neuroimaging leading to conflicting results can be found in the area of the associations between hearing loss and brain structure. There are many studies that agree that hearing loss can reliably predict grey and white matter brain atrophy in elderly subjects and is associated with dementia. However, for this same field, null results have also been found, where no link was found in between hearing loss and brain structure. It is also possible that other null results were simply never published, since there is a lower premium for the publishing of null results in science, resulting in the so-called file drawer effect. Therefore, this is an interesting area to study, because the robustness and the reliability of the result is unclear over different pipelines, datasets, and association measures that researchers are choosing. This project aims to deal with this disagreement scenario by borrowing the idea of continuous integration from software engineering. Now, this idea simply consists of that when a new piece of code is committed to a code repository, it triggers an automatic series of tests that make sure that the code is behaving in an expected manner and generating the expected output. So if we extend this to scientific results, we could, instead of committing code, perhaps commit new pipelines and new datasets and see how they affect the results that researchers are obtaining. Therefore, we want to build an infrastructure that is capable of checking the variability of the results and give researchers a dashboard of the results across different pipelines and datasets in the field. Existing continuous integration uses in terms of scientific results are quite limited in a neuroimaging context. They don't account for the large amounts of data in neuroimaging datasets or for the computationally expensive pipelines, which typically you cannot run on a continuous integration platform. They also lack a generalized or modular continuous integration framework, which makes it very hard for other people to reuse this work. They also don't have a strategy to deal with private or registered clinical datasets that can't simply be uploaded onto a public code repository. Succinctly put, the objective of this project is to create a generic, distributed, in the distributed computation sense of the word, continuous integration framework, which will allow researchers to evaluate neuroimaging results across variations in datasets, pipelines, and processing conditions. We are evaluating this new framework on the associations between hearing loss and hippocampal volumes, and it's yielding a dashboard of results where we will be able to see the associations between every pipeline and every dataset. And this will help researchers ensure the reproducibility of results in their field. And in my case, for example, make sure that these hippocampal volumes and the trends that they produce are robust across pipelines and replicable across datasets. Here is a software architecture diagram for my continuous integration framework. So it has three main modules. I can't go into huge detail due to time constraints, but the first module is the data retrieval module, and it simply aims to ingest datasets from the Canadian Open Neuroscience platform repositories using the DataLad software onto the Compute Canada servers, where I can then make the data available to CBrain, which is the distributed computation system I will be using to launch all of the computations in this project. The second module is the pipeline containerization module, and it similarly aims to make all of these pipelines visible in CBrain. And these are all pipelines that can produce brain segmentations and can produce hippocampal volumes for my use case. The third module, and the one that contains most of my code, is the continuous integration module. And how it works is that the user will fork my project repository on GitHub, and inside it, they will find a user input or experiment definition file where they can specify which of these datasets and pipelines they wish to run the computations on. And every time they edit this experiment definition file, it will trigger a continuous integration run in my continuous integration client, which is Circle CI. And inside this run, it will execute all of my code, which uses the API from CBrain to communicate with CBrain and launch tasks and get task statuses and just make sure everything is flowing nicely. And it stores intermediate steps that make all the results completely reproducible and transparent in a series of JSON cache files. Going and iterating through these cache files allows us to collect the results, which we can then produce visualizations and statistical summaries for inside the continuous integration run, and then output them. So diagrams are very fancy and all, but how is this going in terms of results? Well, I've built a functional prototype of the platform, which includes two pipelines, FSL and FreeSurfer, and one dataset, PreventAD. So I'm going to go ahead and open the link to the GitHub project, and here you will find all of my open code available for anyone to see on GitHub and the complete documentation on how to run the platform. If we click here, we can actually see that it takes us to the continuous integration runs, where we can see the status of the latest computations, and it stores everything for the past 30 days. If we click on the artifacts, we can see the outputs of these computations. And one of these is a JSON cache file, which has all of pretty much it just shows all of the different steps in all of the pipelines and where they're at in terms of computation and the results in terms of hippocampal volumes. Here we can see that it also produced a graph showing left hippocampal volumes versus a hearing loss measure for the FSL pipeline run on the PreventAD dataset, and it has done the same thing for FreeSurfer on the PreventAD dataset. And it's obvious how when I have all of the other pipelines and datasets integrated, this will allow me to construct the dashboard of results. In terms of contributions to open science, one of the main advantages of this platform is that it allows us to ensure the reproducibility of a certain given scientific result with complete openness and transparency. All the information you need to know how a certain result was reached can be found in the continuous integration runs, and this can allow us to consolidate knowledge, quantify and explain the uncertainties of results, and ultimately lead us to having more confidence in what we learn from our experiments. The platform should generalize easily to other neuroimaging domains so that other people can make use of it in their own scientific research, and allows them to systematically evaluate the variability and robustness of results, as well as pinpoint discrepancies caused by pipelines and datasets. It will also very easily allow people to verify results in publications by simply rerunning the computations that they propose with minimal setup. It will allow me to provide the community with immediately reusable derived segmentation data from my hearing loss use case, and it will allow me to produce software, data, and results that will be openly disseminated. The documentation will be made available on NeuroLibre, and any preprints of my resulting research from this will be published on the Open Science Framework. And that is all I have to say today. Here are the references, if in case you wish to take a gander, and thank you for watching.